Hi, my name is Deanna Church, and I've been in the fields of genetics and genomics for about 20 years now, largely spending a lot of my time working on the reference assembly to try and make that available to the community. Over the past year, I joined Personalis because I'm really interested in trying to see how we can better apply genomics to the field of medicine. So today, what I'd like to do is take a little bit of time to introduce you to Personalis and the ways that we think about how genomics can help medicine. So first, I'd like to tell you a little bit about Personalis and how the company was founded. So the company was started by 4 Stanford professors, Ewan Ashley, Mike Snyder, Atul Butte, and Russ Altman, and John West. The impetus behind this was the fact that the interpretation of a healthy human genome sequence by different pipelines would often produce different results. And additionally, the amount of effort that it took to interpret that genome in a clinical setting so that results could be returned to a physician took a lot of time and effort. And so Personalis was started with the notion of trying to come up with the most accurate sequencing approach and analysis that technology allows, as well as trying to think of ways that we could improve that annotation and interpretation process so that it would be more readily applicable to the clinic. So we've taken what we think is a very holistic approach to this, trying to analyze every step of the pipeline in order to figure out where sequencing problems go wrong and how we can improve that process. And we do that all in a CAP/CLIA certified environment so that we can then return these results to patients. Now, I think one of the things that's interesting when I talk to people about genomics in medicine is many people really seem to think that most of the problems that we have with sequencing and analysis are bioinformatics problems. You know, that is, we really just need better software or we need better analysis tools. But the fact of the matter is, when we really start to look at where some of the problems in genome analysis are, they can be at really every step of the pipeline. This starts at the sequencing step. It can involve the bioinformatic pipeline, but also in the interpretation step. And so we've really taken an integrated approach to trying to solve problems at every step of this pathway, and that's led to what we call the ACE platform. And what we can do is we can take this one platform and apply it to many different applications, from large-scale sequencing and annotation, to cancer studies, to Mendelian genetics, as well as pharmacogenetic studies, as well as just looking at disease risk in the population. We also offer a clinical test that sits upon this platform that allows us to return results to patients. So when we think about this whole problem of genome analysis, I think it's really useful for us to think where we've been successful as a field. And largely, this has been in the case where it's a single variant affecting a very highly penetrant phenotype. But where we would really like to be is we'd like to be able to look at the genome of healthy individuals and be able to explain the normal variation that we see within that population. However, even in this area where we consider ourselves to be the most successful, that is in diagnosing generally fairly rare, highly penetrant disease, we don't get the answer right 100% of the time. In fact, we're really closer to about half the time. So what I can show you here is both a representation of how we've been doing when we look at various cases, and that is we're able to return a result to the physician about half the time, versus numbers that we see reported from different labs as they're addressing this. And when that range is roughly from 25 to 56%. And this is not surprising. We know that some of these problems are based on the difficulty in identifying the variant. Some of them are based on the fact that one disease can be caused by variations in many different genes, and in some cases the phenotype is just not well enough described. But when we really look at the problem hard, we can find places along the entire spectrum where we can identify problems. And we tend to try and break these down into 2 categories. So we often think about one problem as analytical validity. That is, what is our ability to really accurately call variants? Because as you can well imagine, it's going to be very hard to return a result to a physician if we can't even identify the variant. Versus the issue of clinical validity. That is, given a list of variants, how reliably can we associate those variants with biologically relevant information? Of course, the crux of the matter is that even though we tend to think about these as 2 different categories, it's really difficult to break these apart when you start thinking about the entire integrated process that it takes to get from your DNA sample all the way through to an annotated list of variants. There are many places in the pipeline where individual problems can affect either one of those issues. So one of the really standard things that we see, and I think this is a problem that the field is trying really hard to address, is that we can see from this graph in the top left corner that even if you take the exact same data, if you run that through different pipelines, you're going to end up getting different answers. And this can happen for many reasons. Part of it may have to do with the analysis alignment that you're doing or the variant calling heuristics that you're using. I think one of the other problems that we've seen when we try to stand up, say, a research pipeline in the clinical setting, is that in many cases these research pipelines just aren't suitable for a clinical setting right out of the box. And that is because, as shown in the graph in the lower right here, these research pipelines are typically tuned to have a low false discovery rate. That is, they don't want to create a lot of false positives and populate the databases with those bad variants, but they do that at the cost of sensitivity. And so it's quite clear if we can't identify the variant, we're not going to be able to return a result. I think the other problem that we see is that whether you're doing a whole exome study or a whole genome study, not every part of the gene or genome is going to be covered. And that can be due to several reasons. So one issue can arise because of genome composition. So for example, we know that regions of the genome that have a high GC content can often be difficult to sequence or even to capture given a capture protocol. And this is one of the reasons that we've actually spent a significant amount of time developing specific sample and library prep protocols to allow us to provide better template to the sequencing machines. Of course, another issue with this can be with your capture reagent. So if you're doing an enrichment sort of protocol, you have to be very careful that you're capturing all the medically relevant content in the genome that you want to analyze in order for those variants to be in your call set. In these slides, we can see an issue with a gene called RPGR. This gene is associated with approximately 80% of cases of retinitis pigmentosa. And we can see that whether you're using a standard whole exome or a standard whole genome approach, the same region of this gene can be very poorly covered. And unfortunately, this is a region of the gene that actually has a clinically relevant variant in it. So we've actually had to work hard both on the sample prep, sequencing, and analysis side to make sure that we can identify that variant in our call set. So I'd like to thank you for taking the time to listen to my brief introduction to Personalis and the way that we think about genomic medicine. If you have any questions about this presentation or any of the services that we offer, please visit our website at personalis.com. Thank you.